Hi everyone, welcome back to the third lecture of this series. Now, sorry it takes a little bit longer time to produce this video because I've been redrawing a lot of things from the book. Now, last week or last time we looked at our cells. We get a first date, so we learn a little bit about the immune cells. This time we're having our second date, so we're going to learn a lot more about these immune cells and as well as their receptors and how they respond to invading organisms or pathogens. So let's get started. So here are the lecture objectives. We're going to look at the concepts very briefly about receptor and ligand binding in the innate system, look at interactions between antigens and different innate immune cells, and also describe those receptors involved in those functions and responses. So here is the recap of what we've learned last week in the innate and adaptive immune system overview of that. Now notice that today we are going to focus on the recognition of different antigens, and we're focused on the innate branch this time. Notice that their recognitions are considered as not specific and mostly based on recognizing pathogen-associated molecular patterns, or PAMPs. Before we look at PAMPs, let's have a review or overview of receptor ligand binding principles. Now, all of the immune responses that we are going to talk about here in the innate and in future adaptive immunity are responses that are initiated between an interactions or binding of a receptor and a ligand. And a ligand we oftentimes consider as a molecule that have interactions with the receptor that is located on the white blood cells in our case. So these interactions will trigger the activations of the leukocytes. And what is more important is that the shape of the ligand and its receptors are critical, and how they fit together determines their binding affinity. So what is binding affinity? So it describes the effectiveness of interactions between the ligand and receptors, and the higher the affinity, the stronger the interactions. And all these receptors that we are referring to, a lot of the time they are on the cell surface. Sometimes they may be soluble molecules that can be either secreted by these white blood cells or even internally within the cell. Now, in terms of ligand, they may be expressed by cells as a cell surface molecule, so it would be a cell-cell touching or cell-cell interactions, or it could also be soluble molecules such as those secreted by the cells. We will talk about things like cytokines and chemokines today. So the binding between the ligand and the receptor will generate some responses, and oftentimes there could be positive signals or negative signals. And these different signals dictates the actions of the one of the cells. Are they going to respond or not respond to that stimulations? In terms of macrophages, is to eat or not to eat. And in terms of NK cells, we're going to look at it is to kill or not to kill. And that is the questions, right? So here is just a picture of me. I had a t-shirt design when I was teaching tuberculosis, TB or not TB. Maybe I can sell that as a merchandise, right? When we are looking at the differences in receptors between different branches of the immune system, in the innate immune systems, they usually have a limited set of receptors that can recognize epitopes that is expressed by a wide range of microorganisms, meaning these epitopes or antigens are commonly conserved between different species of microorganisms. Now, in adaptive immune systems, they can recognize a vast number, a huge amount of epitope that is expressed by different pathogens, and mostly by very specific lymphocyte receptors that are expressed by B cells and T cells. And last time we looked at the different receptors. B cells, they carry immunoglobulins that is on their membrane, and T cells have their specific T cell receptors as well. Now, their recognition is to be considered as very precise, very specific for a given antigen. And in this lecture, let's focus on the innate immune system for now. The PAMPs are the collective names of macromolecules that are recognized by the innate immune system. For example, we have lipopolysaccharides, LPS, oligosaccharides, proteins, lipids, RNA, DNA, peptidoglycans that are very commonly found in different bacteria species. And we have specific receptors on the immune cells that will recognize these PAMPs. Now here, when we talk about specific, it's not like the specificity of the adaptive immune system. Here, we're only talking about a category of specific receptors that can recognize a category of specific PAMPs. And we are not talking about a specific receptor that can only recognize one antigen. Notice the difference. Now, depending upon the particular receptor and ligand and upon the type of cells or molecules that the receptor is associated with, binding between ligand and receptors can provide different events, including, for example, some receptors are designed to bind molecules that then generate signals between cells. Some are designed to detect the presence of intruders in the environment. And lastly, some can also examine their labels, seeing to test if they are belong to self or do not belong to self. Are they a threat or not a threat? In terms of innate immune systems, we have this broad structural motif that can be recognized by receptors that we call pattern recognition receptors, or abbreviated PRRs. Now, there are two types of pattern recognition receptors. A lot of them are on the surface of the cells, and some of them are in soluble forms that can be found in the cytosol of a cell. Now, they can recognize pathogen-associated molecular patterns, and when the PAMPs bind to PRRs, it will trigger various forms of innate immune responses. And the ultimate goal is to destroy the pathogen, or sometimes destroying the cells that are infected by pathogens, namely viruses. So we are going to look at all of these processes in greater detail today. One important thing about these PRR is that they are preformed receptors on the innate immune cells, and that means that we are born with these receptors. Now, unlike the receptors that are specific in the adaptive immune system, we need to learn and generate these new receptors. Now, these PRRs, however, are there all the time, so it can mount a very quick response that fits the definitions of innate immune system responses. Now, here are just some of the examples of the PRRs, such as LPS, lipopolysaccharide receptors, scavenger receptors, toll-like receptors, Mannose glycan receptors, killer activations or inhibition receptors, and complement receptors. In terms of complement receptors, we will look at it in our next lecture when we focused on the complement system. Let's look at macrophages and their receptors. Macrophage, the big eater. Remember, pattern recognition receptors are designed to recognize things that are non-self, meaning they are from pathogens. Now, here are the two receptors, mannose and glucan receptors. Now, they can bind to carbohydrates expressed on the surface of a wide variety of microbes and pathogens. And LPS receptors can bind to sugar or lipid moiety, LPS, that is commonly expressed on many gram-negative bacteria cell walls. Scavenger receptors can recognize negatively charged ligands, particularly phosphate-containing lipoteichoic acid that is present in the cell's walls of gram-positive bacteria. After recognition, bacteria will bind to macrophage receptors, and this will initiate the release of cytokines and chemokines, which are soluble chemicals that can promote inflammation and chemotaxis. Along with the binding process, it will also initiate receptor-mediated endocytosis, meaning there will be phagocytes forming that is engulfing the bacteria. It will also later fuse with lysosomes, which contains cytotoxic contents and such as reactive oxygen species like hydrogen peroxide that can help to destroy the bacteria that is engulfed and encapsulated in the phagosome. When the phagosome and lysosome fuse together, it formed a entity. We call them phagolysosome, which is the fusions of the two. The pathogen is being broken down in this phagolysosome into proteins, lipid, and polysaccharide components. What I didn't show in this figure is that part of these broken down products can also be displayed on MHC class II molecules as an antigen-presenting process for the adaptive immune system to learn about this bacteria or other pathogen that is being broken down. Among all of the pattern recognition receptors, let's focus on Toll-like receptors for a detailed discussion. Now, toll-like receptors are present on a variety of innate immune cells and epithelial cells. When they bind to PMPs on infectious organisms, it will mediate the generations of different defense responses, such as activating a lot of the transcriptional factor and process, which lead to synthesis and secretions of cytokines to promote inflammation. It will also attract more macrophages, neutrophils, natural killer cells, and dendritic cells to the site of infection to mount an innate immune response. Here is just a fun cartoon to show you toll-like receptors that are located on the surface of a cell, which can sense the presence of foreign material. Here would be a piece of perhaps viral RNA or viral DNA. Now, in a biochemical sense, these toll-like receptors do contain two domains. Now, we will focus on the pathogen recognition domains, which is often sit on the outside of the cell membrane facing the environment. Now, here just a table looking at different ligands from different microorganisms. These are all examples that can be recognized by specific subtypes of toll-like receptors. Now, when I look at toll-like receptors, it reminds me something. The horn of a sheep. Out of all of the subtypes of toll-like receptors, I want to focus on toll-like receptor 4 for our discussion. Now, toll-like receptor 4 is found on macrophage cell surface, and they can recognize bacterial lipopolysaccharides, or LPS, commonly found on gram-negative bacteria surface. Now, actually, their recognitions require some work and require assembly and participations of other co-receptors. Now, first we have a co-receptor called LBP. Well, it stands for lipopolysaccharides or LPS binding protein. Now, this binding protein can deliver LPS to co-receptor complex, which consists of the CD14 or the LPS receptor. Now, notice that the binding between LPS and the cell surface, it happens on CD14. But the LPS is sensed by toll-like receptor 4, and this sensing will activate an array of cellular mechanisms and leading to activation of NF-copper B. Now, NF-κB will stimulate the synthesis of different inflammatory cytokines, which will get exocytosed and leading to an array of immune responses. So here are 5 major inflammatory cytokines and chemokines secreted by macrophages that I think is very important, that I have to bring it up. The first one is interleukin-1. Now, interleukin-1 can further activate macrophages, so more will be activated. And the second one would be TNF-alpha. It can increase permeabilities of capillaries. Why? So that we can recruit more innate immune cells to the site of infection. The third cytokine would be IL-6. Now, IL-6 has a very important role in the body, is to increase body temperature, leading to a state called fever. Now, why do we need fever? Well, actually, a little bit higher body temperature can often impair pathogen replications, which is helpful for us to recover. I know, however, fever is not a pleasant feeling, and that's why we oftentimes use medications to suppress fever. Now, the last cytokine that I want to bring up is IL-12. It can recruit and activate NK cells, and we'll look at NK cells' functions and response in the later half of today's lecture. Now, notice that the CXCL8 is separated out. It is a type of chemokine. Now, chemokine CXCL8 can recruit neutrophils from the circulations to the site of infections and leading to more acute inflammation response. Again, we are going to spend a lecture to talk about acute inflammation later. So far, we have looked at receptors and molecules to tell non-self. What about self molecules? Now, here is a very important one called MHC molecules. Now, they are highly variable from person to person, and in fact, there are 2 major classes. First one is MHC class 1. They're expressed on all nucleated cells. And MHC class 2 are primarily expressed by immune cells, mainly on macrophages, dendritic cells, B cells, and T cells. The main purpose is to present antigen. In human, MHC molecules are also called human leukocyte antigens, or HLA. They are used to distinguish the compatibility of bone marrow and organs during transplants, and more about HLA later when we talked about specific topics related to transplants. Now let's look at neutrophils and their receptors. I call neutrophil the ninja assassins of the immune system, and you will find out why. Here is a graphical illustration of the neutrophils. Now notice that they bear receptors that are very similar to those that are utilized by the macrophages. And neutrophils, however, have a relatively short lifespan, about 2 days in the circulations. Now, in contrast to macrophage, when they perform phagocytosis, their phagosome fuse with their lysosomes. Neutrophils have a wider array of more toxic antimicrobial granules. Then they will fuse into the phagosome and destroy the bacteria. The macrophages, after their phagocytosis process, they would do an antigen presenting. However, neutrophils after phagocytosis will undergo apoptosis or programmed cell death, and eventually these leftover neutrophils will be degraded by macrophages. So notice this is the key difference between the phagocytosis process between neutrophils and macrophages. So why ninja? Well, ninja oftentimes sacrifice themselves after a mission, so just like our neutrophils sacrifice themselves after phagocytosis. Well, I know you could argue they could be in other types of warrior. Anyhow, now neutrophils also produce a wide array of chemokines. Now, Again, chemokines are small proteins that guide other leukocytes or white cells to the site of infections, which is an essential part of the innate immune response. Now, here are the three different chemokines that I want to bring you to attention. First, we've seen that already, which is the CXCL8, which can recruit more neutrophils from the bloodstream to the site of infection. Second one will be CCL2, which can recruit T cells and dendritic cells to the site of infection. And CCL3 can recruit granulocytes, such as these mast cells, eosinophil, basophils, to the site of infections. Now let's look at dendritic cells and their receptors. Now I'd call it a superstar in antigen presenting because it is the main bridge between the innate and the adaptive immunity. There are multiple types of toll-like receptors on dendritic cells. First, we have toll-like receptor 2, which can recognize polysaccharides found on gram-positive bacteria. Toll-like receptor 4 that can recognize LPS found on gram-negative bacteria. And we also have toll-like receptor 3 that can recognize viral RNA found within the virus. Now notice that with these three receptors, it can recognize a wide array of pathogenic organisms. Now what do they do after recognition? They can perform phagocytosis. Now notice that they do carry both types of MHC class molecules. Remember, all nucleated cells carry MHC class 1. So, of course, it carries MHC class 1. It also carries MHC class 2, which can perform antigen presentings to T cells. Now let's look at natural killer cells, which they carry immune surveillance, and they often circulate and make cell-to-cell contact with other human cells. Why do they do that? Well, they are going to recognize and destroy viral infected cells and also tumor cells. Here, let's remind ourselves, NK cells are part of the lymphocyte lineage, but they do not have antigen-specific receptors. Now, NK cells do display a variety of receptors on their surface that is capable of stimulating or inhibiting their own cytotoxic actions. Now here is a figure that shows you different receptors that we're going to talk about. Now when it encounters healthy cells, that displays marker that can actually initiate an inhibitory responses to prevent themselves from being killed. So no killing of healthy cells. Now let's take some time to look at how NK cell to perform killing. Now remember, they have receptors that can detect differences or alterations in host cells that have been infected by pathogens, and particularly we are referring to viruses. So here is the mechanisms of how innate immune cells kill or respond to viral infections. Now, this receptor on NK cell, we call it killer activation receptors. So it allows NK cells to recognize the presence of stress-related molecules called either MIC-A or MIC-B. Now, MIC-A or MIC-B are expressed by host cells that are unhealthy or abnormal for a variety of reasons, including being infected, and binding between MIC-A or MIC-B by NK cells' killer activation receptors will induce the NK cell to attach and release lytic granules to destroy the target infected cells. So what about inhibitions? How does NK cell knows when not to kill? Well, NK cells utilize a different type of receptors called killer inhibition receptors, or KIR. They are for monitoring major histocompatibility complex or MHC class 1 molecules that displays on all cells that have a nucleus. Now, this is a method to see if the cell is normal or not. Now, some cancer cells and some type of viral infections actually decreases the number of MHC class 1 molecules displayed on the surface. And this abnormality can be detected or sensed by killer inhibition receptors. Now, if the level is subnormal, NK cells will initiate killing of the target cell. On the other hand, if the level of MHC class 1 is normal, and it will not initiate the killing process, so the process is terminated and the target cells is released unharmed. Well, I sort of want to dress up like Terminator one time. So here is the graphical summary of what we've just talked about. Notice that the binding of normal number of MHC class 1 would lead to killer inhibition and not harming the cell. And binding of stress-related molecules, MIC-A or MIC-B, to killer activating receptors will lead to release of lytic granules that will kill the stressed cells, which are often infected by viruses. And here is the graph to represent virus-infected cells. Now they display these MIC-A or MIC-B. Now these will activate the killer activator receptors, and the NK cells will release cytotoxic granules directly onto the target. So what are these granules? The first one is perforin. The name tells you it creates a perforates. It perforates the cell membranes, meaning it creates a hole and triggers cell lysis. And the second granules that are released are called granzymes. These granzymes will trigger apoptosis or programmed cell death of the viral infected cells. The idea is to kill the cell so that the virus can no longer use host mechanisms to replicate. Now moving on to some soluble proteins. So we're no longer talking about specific receptors, specific cell response here, but we're talking about soluble proteins that have a role in defending viral infection. Notice this is still part of the innate immune response. Now these soluble proteins are called interferons. They're induced by the presence of viral particles. They interfere with viral replications in infected cells, and they also induce resistance to viral infection in uninfected cells. And we're going to briefly look at the mechanisms. So again, interferon is part of the innate immunity against viral infections. So there are several types of interferon, and the type 1 interferon, including interferon alpha and beta, are synthesized when there is a viral infection in the cells. So it triggers virally infected cells to synthesize and excrete interferon alpha and interferon beta. Now these interferons have some role, and let's look at what do they do. Now, collectively, the interferons have three major effects. And the first one is to induce resistance to viral replications in all cells, including neighboring healthy cells and as well as the infected cells. Now, when interferons bind to their corresponding receptors, it will decrease protein synthesis of neighboring cells and as well as their own cells. Now, how do they decrease protein synthesis? It is by interfering ribosomal proteins. So less protein would be available, and also their ribosomal activities would be decreased so that the viruses cannot use these host ribosomes to replicate their proteins efficiently. And second, it will have effect on NK cells. Now it will increase the expressions of ligand receptors on NK cells, meaning that there will be molecular activating receptors to sense these stressed cells and induce apoptosis or lysing these infected cells. And the third function is also on the NK cells. It will activate NK cells to kill more viral infected cells by increasing recruitment of NK cells to the site of viral infections and also increasing their numbers, meaning proliferations. So that is the end of this lecture. Here we have just take a second look of all these innate immune cells and their responses. In our next lecture, we're going to look at the complement system and how it helps us to defend pathogens. So stay tuned.